In this video, we will learn about the drugs penicillins. The history of penicillins can be traced back to the discovery of penicillin by a Scottish bacteriologist, Alexander Fleming, in 1928. He conducted a series of experiments at St. Mary's Hospital in London when he noticed that a mold called as Penicillium notatum, it has inhibited the growth of bacteria, or it had killed the bacteria in a petri dish that he had left unattended. So Fleming, he identified that the mold produces a substance that could inhibit the growth of various bacteria, which he named as penicillin. So this is how the journey of penicillins began. Let's start with the chemistry of the penicillins. So as we know that penicillins, they are beta-lactam antibiotics. We have all heard this word that they are beta-lactam antibiotics. So what does this beta-lactam means? So lactam, it's a cyclic amide. It's a completely chemical term which means a cyclic amide. What is an amide? The amide, it's a chemical group in which a carbonyl carbon in which a carbon is attached to an oxygen by a double bond, and an alkyl group is attached along one side. So if this carbonyl carbon is directly attached to a nitrogen, this is called as an amide. Now if this structure amide is in a cyclic form, it makes a lactam, which means that if we put this amide group, now this is an amide group, if we put this in a cyclic compound, it becomes a lactam. So this is a lactam. Now the carbon that is directly attached to the carbonyl carbon, this is a carbonyl carbon. The carbon that is directly attached with the carbonyl carbon is called as alpha carbon, and the carbon which is adjacent to the alpha carbon is known as beta carbon. So that is why the name beta-lactam came into being. So this is the chemistry of a beta-lactam. So penicillins, they all have a beta-lactam ring in their formula, in their chemical structure. This is the structure of a penicillin, and we can see that here we have a beta-lactam ring. And this ring is common to all the beta-lactam antibiotics. So this is the structure of the penicillin. So this alkyl group here, which is denoted by R, it is changed by a variety of chemical constituents, and the synthetic penicillins, they were formed from this basic compound, from this basic chemistry. Now let's move towards the classification of the penicillins, now that we have understood the chemistry and what a beta-lactam is. So the classification of penicillins. The penicillins, they are classified into three basic classes. The first one is the natural penicillins. It has two drugs, penicillin G and penicillin V. These are the prototypic, or these are the parent drugs of this class of drugs. Now the second class, it is called as anti-staphylococcal penicillins because they have activity against staphylococci and streptococci as well. So the drugs in this class are oxacillin, dicloxacillin, cloxacillin, nefcillin, and methicillin. So these are the 5 drugs that comes in the anti-staphylococcal penicillins. They have activity against staphylococci, as the name indicates, and they have activity against streptococci. The penicillin G and penicillin V, the first class, they have activity against gram-positive bacteria. They have activity against gram-negative cocci. And they have activity against anaerobes, which does not produce a beta-lactamase. So this is the spectrum of penicillin G and penicillin V. Now the extended spectrum penicillins, as the name indicates, they have an extended spectrum of activity. We have three subclasses of the extended spectrum penicillins. Number one is the aminopenicillins. It has ampicillin and amoxicillin. The second class is carboxypenicillins, which have dicarcillin and carbenicillin. And the third class is uridopenicillins, which have piperacillin, meslocillin, and eslocillin. You can make mnemonics in order to remember the drugs, but the names that you have to remember. Then that in the extended spectrum penicillins, we have three subclasses. And now they, as the name indicates, they have an extended spectrum of activity, which means that they will have the activity of penicillins, the natural penicillins, and along with that, they have activity, or they have more pronounced activity against the gram negative bacteria. And this activity or this property is unique to this class because the previous classes of the penicillins and antistaphylococcal penicillins, they do not have activity against gram-negative rods. So the extended spectrum penicillins, they also have activity against gram-negative rods and cocci as well. They also have activity against gram-positive bacteria. So they are more broad spectrum, or they're more extended spectrum penicillins, as the name indicates. Now let's move towards the mechanism of action by which the penicillins work. So now here we have a cell wall. We have a structure of cell wall here, and this is the cell wall of a gram-negative bacteria because it has these porins. So now we can see that this right structure right here is the cell wall, and after the cell wall there is periplasmic space, which have the penicillin binding proteins, and then we have the cytoplasmic membrane, and after this the cytoplasm of the bacterial cell is present. So in order to understand the mechanism of action, we must have a sound knowledge of bacterial cell wall. So in the bacterial cell wall, this structure, which is the peptidoglycan, it is the basic structure on which the penicillins act. Now, what we are going to do is that we are going to take a part of this peptidoglycan and we will move it towards another slide in order to better understand that how the penicillins actually work. So it blocks the transpeptidation reaction in the polypeptide. So this peptidoglycan in the bacterial cell wall, it actually acts as the backbone of its cell wall, and it is responsible for maintaining the integrity and cell shape, and it also prevents the cell lysis from high osmotic pressures. So this peptidoglycan, it has two constitutes. It has a polypeptide, which is a ring, or which is a chain of amino acids, and it has a polysaccharide, which is a polymer of carbohydrates or sugars. So these two proteins, so this is a protein and this is basically sugar. So these two, they combine and they make the peptidoglycan, which is the backbone of any bacterial cell wall. So the bacteria or the antibiotics, the beta-lactam antibiotics, they actually act on this peptidoglycan and they block the synthesis of peptidoglycan, and they block the transpeptidation reaction that is very crucial in the synthesis of peptidoglycan. So in this peptidoglycan, that we have already discussed, that it has a polypeptide, which means it has amino acids, and it has a polysaccharides, which are basically sugars. The structure of this peptidoglycan is that it has alternative sugars. They are connected to each other. So they have two sugars, which are alternating. One is N-acetyl muramic acid, and it is also known as NAM, and the other one is N-acetyl glucosamine and is also known as NAG. So this is N-acetyl muramic acid and this is N-acetyl glucosamine. And these sugars, they are, I'm going to write M and G. So M means NAG, NAM, sorry, and G means NAG. So these sugars, they are connected to each other and they make the whole polypeptide. So the polypeptide that we were talking about, this is the polypeptide that we were talking about. So to this M or to this N-acetylmuramic acid, to this sugar moiety, the chain of amino acids is usually attached. So this is the chain of the amino acid that is usually attached to the N-acetylmuramic acid, which is a sugar moiety, which is a sugar in the polysaccharide unit. So here we have the proteins or the chain of amino acids. So this chain of amino acid basically connects with the other chain of amino acid of another sugar chain. Now we have two sugar chains and between them is a bond. This is known as transpeptidation. Now what penicillins does, that they blocks this transpeptidation reaction. Now when the transpeptidation does not occurs, as a result, they inhibit the transpeptidation reaction, they halt the peptidoglycan synthesis, and as a result, the cell dies. And the exact mechanism of cell death is not completely understood, but autolysins and the disruption of cell wall morphogenesis are involved. So beta-lactam antibiotics, they kill bacterial cells only when they're actively growing. So this is how they block the transpeptidation reaction. I'm going to recap it, that we have in the peptidoglycan, it is the backbone of the bacterial cell wall. In the peptidoglycan we have two moieties or two components, a polypeptide, which is a chain of amino acids, and we have a polysaccharide, which is a chain of sugars. So in the structure we have, these are the sugars, and along with these sugars the amino acid chains are attached, and the amino acid, one amino acid, it is attached to the amino acid of another chain, and this process or this step is known as transpeptidation. So what beta-lactam does, that it blocks the transpeptidation reaction and it results in the death of the bacterial cell wall. So this is how they have their antibiotic or they have their antibacterial effect. So let's discuss the pharmacokinetics. Pharmacokinetics basically it includes the absorption, distribution, metabolism, and excretion of the drugs. So the absorption of orally administered drugs, they differ greatly for different penicillins, and it depends on their acid stability and their protein binding. So both these factors are very important when we are discussing the absorption of the various penicillins. So GI absorption of some drugs are erratic, so they're not suitable for oral administration and they are given in the form of IV injections. So we have very few drugs that can be given through the oral administration, and these are ampicillin. We can give it in the oral formulation as well. We can give amoxicillin, and we can give diacloxacillin. So these three drugs, they can be given as in the oral formulations, and apart from them, the rest of the penicillins, they have to be administered by the IV or IM route, because they're not stable and they are degraded in the stomach and they does not reach the blood, or they does not reach the serum concentration that is effective to kill the bacteria. So absorption of most oral penicillins, it is impaired by the food, so they must be taken 1 or 2 hours before or after the meal. So the IV administration of penicillin G, it is usually preferred to the IM formulation, because the IM formulation, it causes a lot of pain at the injection site. Now, the distribution. They are very widely distributed throughout the body because these are polar molecules. So the intracellular concentration, they are well below those found in the extracellular concentration. So the benzathine penicillin and so the concentration in most tissues are equal to those in the serum. So penicillin is also it is also excreted in the sputum and milk. So if we are prescribing penicillins to any mothers who are lactating, we must consider the fact that they are going to be excreted in the milk, and they will reach the baby as well. The metabolism, now that the concentration of penicillins, they're not very well in the CNS and the prostate and the eye. So in these three regions, the concentration of penicillins are not very good. So not good distribution. However, in the situation of inflammation or in the situation of meningitis, the concentrations are good as compared to when the meninges are not inflamed. So they're metabolized in the body by the liver, and the excretion of penicillins, they are primarily excreted by the kidneys, and very small amounts are excreted by the other routes. So the primary mode of excretion is the kidney, and in the kidneys 10% is by glomerular filtration and 90% is by tubular secretion. So any drug that inhibits the tubular secretion of penicillins is going to affect the excretion of the penicillins as well. So the normal half-life, it is approximately of penicillin G is approximately 30 minutes, but in the renal failure, it may be as long as 10 hours. So we must consider the excretion or the renal status while prescribing the doses of penicillins. So there is only one drug that is excreted by biliary excretion, and that drug is naphthalene. It is excreted in the bile by the biliary excretion.